Blue, green, brown. Imagine you having the option to decide what your baby's eye color would be before they were born. Imagine it could go so much further. Embryonic genetic engineering is the future, and it is so much more than just the mere appearance of your baby that can be altered. Any parent given the opportunity to provide their child with a healthier life would take it in a heartbeat. This can be achieved through embryonic genetic engineering when used therapeutically. It can remove genes that are linked to certain defects and disease. It is a moral obligation we have to humanity to ensure the survival of the population. Gene editing is the basis to change our species to be greater and less susceptible to diseases and other genetic abnormalities. Scientists have been using methods such as CRISPR-Cas9, Sanger sequencing, among other methods, to continue to conduct research and therapeutic genetic engineering on the embryonic level. One method that scientists use at the moment is pre-implantation genetic diagnosis, PGD for short, and a termination of pregnancy, which helps to ensure that couples have a healthy child using the moral imperative by G.J. Anais. During this method, multiple embryos are harvested and tested to determine healthy ones, and only those healthy ones are what would be kept. This method helps to prevent against diseases like cystic fibrosis, thalassemia, and Huntington's disease, to name a few. Unfortunately, there is a limit of the amount of diseases that can be prevented with this method due to a small amount of embryos. So there's only a certain amount of embryos that can be harvested during this method. And as a result of these constraints, polygenic diseases such as schizophrenia cannot be weeded out unless the number of embryos increases to a number of 100 of thousands per GJNIs. Another issue that arises using the PGD method is due to the limitations of this method, even with the selection of healthy embryos, unfortunately, the gene in question could still be carried by the embryo and eventually passed on through the gene line. So essentially, the gene is not eradicated altogether. With gene editing, scientists can completely eradicate mutated genes, preventing certain diseases from not only affecting individuals, but totally eliminating it from being carried and passed on to future generations. So, scientists use CRISPR-Cas9, Sanger sequencing, short for clustered regularly interspaced short palindromic repeats, CRISPR associated protein 9, easily remembered as CRISPR-9, in order to obtain this. This method is accomplished by editing stems from an enzyme, which is retrieved from a bacterial antiviral system, and that makes DSBs, also known as double-stranded breaks, at the DNA level, which we all know as deoxyribose nucleic acid, and these sites are complementary to a guide RNA or ribose nucleic acid, and also have a nearby protospacer adjacent motif or PAM sequence, as said by Chris Takibana. So I'll explain that in layman's terms. So that means that bacteria from an antiviral system, which exists in bacteria or archaea, is what is used to slice the area of the DNA that has the disease or problem gene, and it is then removed. The area will then be filled or replaced with a new gene strand that will eliminate the defective disease gene, eliminating and then ultimately eliminating the possibility of the gene to be passed on to any future generations. CRISPR right now is the most commonly used method of gene editing therapy. So now that we've covered the how and you can understand the process and the most commonly used methods today, I'm sure you're still thinking, what about the moral compass? This is still, ethics really come into play here because essentially scientists are experimenting with human life. To ensure a healthy future for the human race, we must find a way to eradicate ourselves of life-threatening and debilitating diseases. According to G.J. Annis, 6% of all births result in some form of a birth defect. 35% of all deaths are due to chronic disease. like diabetes and cancer, just to name a few. So imagine a world where we change these numbers for the positive and therapeutically use embryonic genetic engineering in order to eliminate ourselves of these diseases. One way that scientists eliminate the argue of testing on human embryos is by using a tripronuclear, or 3PN, zygotes to carry out their research. These zygotes have only one oocyte nucleus and two sperm nuclei. Essentially, what that means is that ultimately the embryo would fail to develop like a normal zygote and it would not be viable for implantation into the uterus. These zygotes are incapable of producing an actual baby. So, using the CRISPR method with these zygotes eliminates the chance of the embryo resulting in a live birth of a baby and also removing the negative ethical beliefs and the risk of having a possible birth with major genetic defects or complications. So, scientists can conduct their experiments without creating and killing embryos for research, and the ethics of killing human life for research would be removed from this scenario altogether. I agree, scientists should not have full reign to do as they please. However, with the right regulations and laws, their work on gene editing and genetic therapy can continue while being as moral and ethical as possible. When you think of a genetically engineered embryo, you think of a designer baby, which usually elicits thought of a future where vanity is at the forefront, parents having the ability to design their children's skin color, hair color, eye color. But as you can see, embryonic genetic engineering is so much more than that. That's just scratching the surface of what embryonic genetic engineering used therapeutically can truly do for the human race. As long as the design is focused on genes and not just the mere appearance and gender of the baby, therapeutic genetic engineering can be a real positive for our human species